Hello, our study group Michelle, Deng, Oscar, and Ying are doing a presentation on cellular respiration and fermentation. So the purpose of cellular respiration is to make ATP for the cells, enabling us to perform every kind of work. Without ATP, we would not be able to perform the necessary tasks. Now let's explore the components of how cellular respiration actually works. Glycolysis is the first step in cellular respiration. Each step within glycolysis is catalyzed by an enzyme. Glycolysis has two main stages, its energy investment phase and energy payoff phase. In the investment phase, ATP is used to transfer a phosphate group producing ADP. At the end of the investment stage, it produces two different 3-carbon sugars that are interchangeable. In the energy payoff phase, 2-NAD enters and NAD plus and 2H plus is produced. In the end, 4 ATP, 2 H2O, and 2 pyruvate molecules are created. When talking about the net gross produced, 2 ATP, 2 NADH, 2 H plus, and 2 HO are produced. The process after glycolysis is pyruvate oxidation. In pyruvate oxidation, a carboxyl group is removed from pyruvate and released as carbon dioxide. The 2-carbon molecule from the first step is oxidized, and NAD plus accepts the electrons to form NADH. The oxidized 2-carbon molecule, an acetyl group, is attached to coenzyme A to form acetyl-CoA. Both glycolysis and pyruvate oxidation make up the substrate level phosphorylation.
In the presence of oxygen, pyruvate enters the mitochondria to oxidize. Enzymes remove CO2, creates NADH from NAD+. The product is acetyl-CoA. Acetyl-CoA enters the citric acid cycle combining with oxaloacetate to produce citrate. Molecules of NAD+ are reduced into NADH and H+. CO2 is released. Succinyl CoA loses the CoA and becomes a succinate. ATP or GTP formation takes place. Inorganic phosphate is added. Electrons are transferred into FADH2. Malate is oxidized into oxaloacetate. The cycle repeats again. For each acetate group that enters the cycle, 3 NAD+ are reduced to NADH, 2 ATP are produced, 2 CO2 are released, NADH and FADH2 transport high energy electrons to ETC.
The electron transfer chain is a collection of molecules embedded in the inner mitochondrial. It exchange of multi-protein complexes numbered 1 through 4. NADH gives 2 electrons to the first protein and becomes oxidized to NAD+. FADH2 gives 2 electrons to the second molecule in the chain and becomes oxidized to FAD. Electron carriers switch between the reduced and oxidized forms. Each protein has a prosthetic group. And as the chain goes down, it becomes more electronegative until the most electronegative being the final electron acceptor. The cytochrome are the iron-containing proteins that are between the protein Q and oxygen. The final electron acceptor is oxygen, and along with the H plus ions would make H2O. NADH produces more ATP than FADH2. Chemiosmosis happens next. H plus ions are being pumped across the membrane as the electrons are moving between the proteins. H plus ions need to fuse back to the mitochondrial matrix and goes through the ATP synthase for that process. The H plus ions entering the ATP synthase causes the spinning of the ATP synthase and generates the proton motive force. The energy force allows the ADP plus the phosphate group to create ATP. The end result is 26 or 28 ATPs produced.
Fermentation is the process of making ATP without the use of oxygen. There are two types of fermentation: alcoholic fermentation and lactic acid fermentation. In alcoholic fermentation, sugar is oxidized into two pyruvates. The two pyruvates are then transformed into two acetaldehydes with the release of CO2. During the process, two NADH and two H plus are used to make two NAD plus, so that the two NAD plus can be used in the glycolysis again. The end product of alcoholic fermentation is 2-ethanol. Also, ADP and phosphate inorganic are used to make ATP, and 2-ATP are mixed during the process. In lactic acid fermentation, pyruvate is reduced directly by NADH to form lactate. There is no release of CO2. This process is sometimes used to make cheese and yogurt. It is also released by muscle cells in the body. And the process produces ATP by enzyme substrate phosphorylation.